Oops, that's not a good start. And so we're going to begin our cardiovascular chapter, and we're going to start with blood. We already have a general idea of what blood is during our tissue exam. What was the proper name for red blood cells? Does anybody remember? Erythrocytes. And what's the proper name for white blood cells? Leukocytes. And so the cardiovascular system is made of the heart, blood vessels, and blood. And when you think about the heart itself is the motor behind all the motion of the blood in the blood vessels. And so it's situated between our mediastinum and mediastinum, and that's going to be the center cavity where your heart is located. Pretty sure I said that wrong. And blood vessels now you're going to have arteries and you're going to have veins. Arteries use the A to remind you that it carries blood away from the heart. Arteries carry blood away from the heart. The veins return blood back to the heart. And try to think about in the major context that the role of blood is to deliver oxygen to all the tissues, right? These muscles here that we're going to learn about in lab next week, they need oxygen. So your lungs are located here. You can see that there's two locations right there. And then you're going to see a lot of times you're going to see this mix of blue and red, which implies the capillaries. See this capillary network. You can see it in other locations. This one's a little more obvious. And so what you have to imagine to yourself is that this is exchange sites for tissues, meaning your liver sits right here. So whatever network this is serves the liver. Here in this segment here, you can encounter at the very back of the body, speaking simply. You can find your kidneys and the red ones, right? They're going to deliver blood away from the heart, meaning that it's oxygenated now. And it's going to deliver it to that organ. And to that same, the other side of that story is once blood has arrived to serve that organ, right, blood delivered that oxygen, it then picks up CO2, which is a waste produced from the use of oxygen, and then your red blood cells have the duty to return it back to the heart. Because the heart's job is to deliver things between the pulmonary circuit, meaning into the lungs, right, to get oxygenated, and then to actually send it out to deliver that oxygen and return it back so that it can get reoxygenated. So we're going to learn a little bit more about blood flow next week when we get back, when we learn about the heart. But to end, we're going to learn about arteries and veins at that same day. And then we're going to see exactly what capillaries are when we zoom in. But today what we really care about is blood. What is actually inside of those blood vessels? And so some functions, right, the most obvious example now at this point is that it delivers oxygen, and also it transport carbon dioxide back to the lungs so that you can breathe it out and get rid of it. This is considered a waste gas, and this is a gas that your cells need to carry out their metabolism. Nutrients, right, we know that when you consume foods, they go through your digestive tract. And in some instances, it needs to be delivered of areas where there's adipose cells so they can store it and form more fat within your body and store those nutrients for when you need it. And nutrients, again, going to be traveling with the blood. Hormones, these are secreted by glands, and I can't really think of an example we've really gone through in class yet. We haven't really gone over any hormones really at this level. But the glands we have, made of cuboidal cells most of the time, and they secrete substances, and then they end up somewhere. How do they get there? They use your cardiovascular system, right? You have glands up here, the pituitary gland, which is going to come up in about 3-4 more weeks. And that pituitary gland controls the organs of reproduction. I think we've talked about that example before. And they get there through the blood. And also waste. So with nutrients and waste, right? You have to think about when you consume anything, eventually you're going to have to defecate whatever you didn't use or whatever you couldn't use, and then it's going to exit out in the form of feces. So if you think about each cell, right? Because at this point, you've kind of seen this is more of a cell bio class, it's felt like. And every single cell, whether it's a muscle fiber, whether it's an epithelial cell, whether it's those epithelial cells, right, squamous, cuboidal, and columnar cells, they all require nutrients. They require carbohydrates, they require lipids, they require nucleic acids and proteins. And so once you consume them, the role of your digestive tract is to place them into circulation. And then it'll deliver all those nutrients or to the liver to be processed, and then it will be delivered. So your blood vessels are like a highway for blood, and the delivery drivers for oxygen are going to be blood, and the delivery drivers for oxygen are going to be blood and also for carbon dioxide. But nutrients and waste are not interacting with blood. They're traveling through, what's the fluid matrix of all blood cells called? Starts with a P. Plasma. And so they're traveling in the plasma. And as a reminder, maybe because I'm still a little hungry, but if you think about a bowl of soup, right, you have your liquid broth, and that's can be considered the plasma of a soup. Like, think about a soup. The liquid component is the plasma, and then the red blood cells, they're all swimming within it. So they're not part of the plasma. You can separate all the red blood cells and all the white blood cells. I'm just going to change I was going to use white here. And then let's pretend these are white blood cells. You can remove all of them and be left with the plasma. There's a company called Grifols, and if you're interested, you can donate plasma and they pay you, and if you go regularly, you can just go once a month, I think, and they pay you pretty good actually to donate your plasma. And they extract a lot of things that we're going to learn about today. Yeah, so if any of you are looking for a nice little side hustle. All right, so the functions of blood now. You have the pH regulation. There are sensors within your body that are regulating that fluid's pH as it's flowing through, and they're actually determining if it's the right acidity. If you're not sure what pH is just in reference to how acid or how base something is. Let me write that down. How acidic or how basic. The pH alone sits at around 7.35 to 7.45. For all you future nurses, this is a magic number that you need to know. You need to know this. And anything that shifts the body's pH into this direction or this direction will cause some diseases. So our blood has the responsibility of regulating it. We'll talk about this a little bit more closer next week. And when we get injured as well, we all see that. We've all had a scrape or a cut, and then we have our platelets coming to the rescue to form blood clots. And platelets will come up a little bit later, but that's where this sort of segues in. And that's how we get clotting. So when we say restrict fluid loss, that little scab that you have is to prevent any more blood loss, hence any liquid that's in the plasma, which is more than likely water. It defends against toxins, right? We talked about why blood cells play a role in immunity. We'll get into what each one does. And then the word antibodies. I want you to write here, this is a protein. And I also want you to write globulin ahead of time. Globulin. And it also stabilizes body temperature. When you're cold, right, if you start running, you start walking uphill, you'll notice that your body temperature gets warm. You don't really feel as cold, say if you were running in cold weather. And then also, it can increase the flow of blood in your body, or your heart can decrease or increase. And so if it decreases, you can expect that it's because your body temperature might be too low and it's trying to stay warmer. And if you have a higher body temperature, it's more than likely that your heart is beating way too fast or faster than normal. That's why we feel really hot when we exercise, right? And so these, at a glance, are just these same points that we talked about above. They're just sort of written for you in a sort of a thematic way. Now the fun part. So when you look at blood, I mentioned that soup example. Our plasma is the fluid component, so you can see that water is going to be a major component of it. We're going to have plasma proteins and other solutes, and that is 55%. You want to know this value, 55% plasma. The last 45% is going to be something we refer to as formed elements, but don't let this word confuse you. This word just implies the cells that you already are slightly familiar with, like red blood cells, white blood cells. And the reason they're called formed elements is because sometimes they don't really resemble or have the same function as other cells because they're missing components. Like, for example, red blood cells, they don't have a nucleus. They can't synthesize proteins up to a point. And so they just sort of become little zombies that just migrate, carry around oxygen, and they don't do any cellular activity within them once they've matured. And so they're not really considered cells for that reason. White blood cells, you're going to see that there's some of them that they do so many different things, and they're just, again, considered, they don't have the same components as all cells. So we just refer to these cells as formed elements. And then platelets are cell fragments. And write this in now. It'll come up again, but we want to just sort of get acquainted with this. Cell fragments. Recall that that blood is a fluid connective tissue. It connects multiple organs together by sharing or delivering those nutrients, gases, hence it's connecting them together. Plasma can be described now as our liquid matrix. Remember that the word matrix means the area around, and so the liquid area is the fluid that is in blood. Formed elements, these are going to be, again, those cells and cell fragments. And keep in mind, I don't know if any of you have been to a party recently. We've all seen the 2-liter bottles that you can get at a restaurant, right? The 2 liter of Coke, right? That bottle is 2 liters. And so if you think about in perspective, 4 to 5 liters, that means that's roughly 4 liters would be 2 of those. That's how much blood, on average, when you calculate the overall blood quantity in females, is from 4 to 5 liters. And then for males, it runs from 5 to 6. And this is typically just because of just body size, not really any other reason. And our plasma, again, being 55%, it's going to resemble interstitial fluid. Now, what is interstitial fluid? We've discussed the term, and I'm going to have to draw a little bit. So unless I have something ahead that I can use. No, I do not, but actually I have something older. Let's get it out. 1 second, and... Oops. And none of these will... Maybe this one will. Nope. Oh, right. I know where it is. This. I just opened the same one, didn't I? Okay. I'll just draw it myself. And so, when we look at blood vessels, here's red, here's blue, and then we're going to make it very simple. This is supposed to represent an exchange site, just like what we saw on mine. I know that yours doesn't have color, but when you see this, you have red, and then you have blue. And so these tunnels or these passageways, these lumens of the blood vessels are continuous. Like if we magic school bus our way in here, we'd be able to travel from the arteries and then enter the veins. But the exchange happens in these spaces around, meaning that if the cells around it, the organs here, if they need oxygen, let me get a smaller pencil here. If they need oxygen, it delivers it out of these spaces, delivers it out. And so keep in mind what's flowing in through here is red blood cells, plasma, and all the contents of plasma. And so oxygen can escape the plasma, and then it goes to the fluid that's around that organ. All of this, you have to imagine, is sort of like a wet network. Like it's all fluid exchange. And so when you see this segment here, again, where red blood cell, we travel from the red here to the blue here, from an artery to a vein. But these spaces out here, this is where the tissue is expected to grab its oxygen when it's delivered. So red blood cells they're delivering their oxygen. Oxygen will leave the capillary, that blood vessel, and now oxygen will be in the interstitial fluid. The interstitial fluid is just the space outside of the capillaries, the space that's going to serve the organ in question. And remember that every organ, for the most part, has a capillary network. That's how everything gets delivered. There's like drop-off sites. And so that drop-off sites we refer to as interstitial fluid. And the word itself means the fluid around tissues. The fluid around tissues. And this is where exchange occurs. And we'll talk about all these little components of plasma in a sec, but that's where exchange occurs. And anything to add here? When we say something is a dissolved protein as well, that means that it's floating in the fluid. Because there are some proteins that they become structural, they become more fibrous, and then they sort of stay in place. But to say that a protein is dissolved is to say that it's going to move with the fluid in this instance. It's going to save you a lot of thinking later on in physiology. But when you're thinking about the fluid, it's just proteins that can move with the fluid. Think about it very simply. And now the hematocrit. When you look at this test tube here, if you look at this test tube and we spin it really fast, we centrifuge it, that means to grab it like with these, put it into a machine that pretty much does, have you ever been to that fair and those high school carnivals that spin around, right? They get you sick and make you throw up in front of everyone, that one. And then so that, if you do that to blood, it'll actually split the components into two parts. You'll see that there's going to be the red blood cell component, which is going to be the massive one. And then you're going to see, I'm going to try to do it in a golden straw color here. Oh, that's too bright. And then the plasma, the fluid will separate completely out. And then you'll end up with the white blood cells, they all group up as well. So this is, you can separate these components out. And so this word, some of you will encounter it again. Just keep in mind that when you go from this having whole blood to spinning it down and separating the components, that's called a hematocrit test. But in our context, for us and what matters for our lecture, is that it's just referring to what's the percentage of plasma to red blood cells, or it's really talking about what is the components of whole blood. And so, do I need you to know the percentages? You don't need to know the percentages for the hematocrit. And all I need you to know is just what is the cell content, the percentage of whole blood that is formed elements. Okay, that's what this phrase is badly trying to say. All right. And so this is going to be the majority of your memorization for this component. And follow this flowchart very slowly and one at a time. There'll be a little bit of reading for each one. And then you can fill in any little bit of information that you'd like in here as you study. And so, starting with plasma being 55%, you have plasma proteins, other solutes, and look at this percentage here. Plasma being 55% of a sample of blood, 92% of it is water. You already know what most of it is. Now, the last 8% is what's going to give you a little bit of trouble. You have albumins, globulins, and fibrinogen. And then you also have things like enzymes and hormones. And hormones can be proteins. Enzymes are proteins. And so plasma proteins, again, the word proteins, these are all proteins. They're all made of amino acids. They have peptide bonds between them. And so let's move forward. Then we look at our other solutes. We have things like salts that I always say commonly as salts, or ions such as sodium, potassium, calcium. All of those are electrolytes. And then we have the organic nutrients that you consume. You can see that it has nothing to do with cells. These cells do not carry around those nutrients. Those nutrients are typically either going to be carried around by enzymes or they might be free floating. It just really depends. And then also, if our cells consume food, right, they consume the nutrients, they're also going to produce a waste right back into those blood vessels. And so albumins, being the most abundant, at 60%, we'll learn these percentages. Make sure we know this. Make sure we know globulins is 35%. And there are two categories of globulins. There are transport globulins. They do exactly as I mentioned, is that they carry things. They can carry things. It's in the name. They transport things. It's things aren't going to be free floating in the plasma. They require some protein to grab it, and there we go. Some protein to grab it and to take it to its destination. And other ones, this one's a little bit more relevant. This is going to be antibodies. Antibodies are what provide defense. And so I'm going to give a little talk on it because, we're at a level where there's a lot of information about, bad information about vaccines. And I'll give you all the premise. And all vaccines are not going to function in this manner. But for the most part, it's the best way to understand antibodies. So let's say that we get something dangerous, right? This is a little dangerous little critter here. This is a pathogen, a virus maybe, could be anything. And it enters your body and it will cause you to become sick. And so the thing is, what vaccines are, is that we, as scientists in laboratories, what we do is that we fragment, and this is just going to be an example of a way that we make it. There's many ways to make vaccines, but this is the easiest one, is that you destroy it. You break it apart, you destroy it into little pieces, right? And then you introduce this into the body, but you tell yourself, why would you introduce those dangerous things into your body? Altogether, this organism requires itself to be intact. It can't just infect you if it's not fully functionally together. And so what we do is that we introduce some semblance of the creature. And so what your body will do is that it will read it, it'll determine that it's foreign, and then it'll create what I just like to think about like little drones. Oops, that's not a right way to draw that. Let's see. It would create something that's like a little drone that all they're doing once they're made is that they're always going to remember this ID, right? The ID of something dangerous. And when you look, and when the real thing comes into your actual bloodstream, then you already have something that will recognize it, bind to it before it can cause an infection, and then it'll target this for destruction, because that's all these do, is that they just gather around things like this. Once they detect something dangerous, they'll gather all around it, and then something will come, see that it's marked to be destroyed, and then it'll be completely destroyed before it can cause a systemic or a body, infect your body. A systemic infection. And so that's the premise behind what antibodies are. They are just little proteins that attack pathogens. Ask away.
Do antibodies go away?
Yes, they do. They have a shelf life, so to speak, in the sense that when you have it, it really depends on the antibody. And I wouldn't be able to give you any specific examples, and I don't remember what the difference is, that why some last longer than others. Some of them are just easier for your white blood cells to create, which we'll get into as to who makes these antibodies, but it's white blood cells that make them, called leukocytes. And so it just depends on the antibody. So I can get back to you on that because that's a good question. I want to know the answer to that. And then so next we have fibrinogen. We have 4%. Is the last bit. And this is involved in blood clotting. And now this word, insoluble. This is the word that I was meant to imply that there's a word, right, to say dissolve means typically that something is soluble. So when something is insoluble, that means that it's not really capable of flowing through the fluid. If fibrinogen is in your body, it's not really flowing through. And doesn't that make sense that if you have, say, an injury, right? Let's say we have an injury here. And fibrinogen, see, if we look at this word here, form large insoluble fibrin strands. Insoluble fibrin means that it no longer moves. So when fibrinogen is commanded, directed to form a clot. It's no longer really going with the flow. It's sort of stopping in place, right? This insoluble form, fibrin, is what allows you to form a blood clot. Right? So that's all this means to say. This can be a confusing phrasing. And that's really it. I don't know why I wrote this here. Feel free to get rid of that. And so with that, so we finished our plasma proteins now. Other things in our plasma is our electrolytes. We've discussed sodium, potassium, calcium, magnesium, right? This one's very important. We're going to learn this one today, bicarbonate, because I want to talk about how we get rid of it in our kidneys when we get to that chapter. This one is bicarbonate. And then we know sodium, we know potassium, we know calcium. This one is magnesium, and only those 5 are the ones that I want you to know. So circle those and make sure we know those. The nutrients that we consume, lipids, carbohydrates, and proteins, all of it will be used by your cells to make ATP so they can carry out their functions. And once they've used or carried out those functions, then we produce a waste. And so now this brings me to our kidneys, right? Like, no, we're not talking about urinary just yet. But I want you to think about now that your nutrients were delivered to cells everywhere, but your cells don't have a way of getting rid of that waste. So where do they place it? Right back into your blood vessels. And as it's traveling through your blood vessels, how do you get rid of that waste? And the answer is your kidneys. That's what urine is. Urine is the removal of the waste that your cells create from using those nutrients. Try to hold on to what the kidneys actually do, even though we're not there. But you can see that there's 2 different types of waste now, right? There's urine, and then there's defecation or feces formation. What's the difference? Urine is clearing out your blood of all the waste, and feces is, if I ate this pen right now, you can expect that it's going to come out through that other side, because that's what feces is. It's sort of connected to your digestive tract, gets rid of everything that you couldn't digest or any excess, and then gets rid of it. And then once those nutrients are in your body, it has to be delivered to the cells. It's delivered to the cells, and then the waste go into the blood vessels, and urine is there to remove all the waste. And the common waste that we find in blood is urea, uric acid, creatinine, bilirubin, and ammonia. Make sure we know these. Yes, because, we all from experience, right? We go to the restroom, we probably do both the number 1 and number 2 at the same time, but they're not related at all. So with that, our formed elements, we have platelets, and again, they are cell fragments. They only make up about 0.1%. They look like tiny little specks on microscope slides, and they're considered cell fragments because they break off of larger cells, like large, huge ones. And so they're like little pieces of cells that do something. And what do? They form blood clots. And then we have our white blood cells that are going to vary. We have approximately 5 classes. We do have 5 classes. And within those 5 classes, sometimes there are different individuals within those classes that do different things but sound the same. But fundamentally, 5 different classes. The word leuco is Latin for white. And so this word means white cells. Erythro, this is going to stand for red blood cells. Red blood cells. And red blood cells transport oxygen. That's why, what is a leukemia? Cancer of what? White blood cells. All right. Now the hard part. Let me just check where we're at. Time. Nice. Now this pathway. Let's start at the top. Remember that in bones, I said that we were going to bring this back, and this is what I decided to move or keep it into this, or sort of fuse it into this chapter was, what is red bone marrow, what is yellow marrow? I really wanted to talk about it during the bone exam, or the bone material, but I thought it's already excessive, and it fits better with blood material. And so if you look what red marrow is, yellow marrow is nothing but adipose tissue. But red marrow is where red blood cells are made. So these are called hemocytoblasts. These are the stem cell population that will give rise to every cell that we're going to have to become acquainted with. For example, red blood cell, the platelets. I know that we haven't discussed these yet, but these are all the white blood cells. Lymphocyte, monocyte, neutrophil, eosinophil, and basophil. These are all the white blood cells. We have to know what they do. And so this is a story of how they come to be. And so if you look at hemocytoblasts, you can see you have lymphoid stem cells and you have myeloid stem cells. These lymphoid stem cells, let that remind you of the fact that they're going to turn into lymphocytes. Lymphocytes. Now, what do we need to know as students? You are free to cross this one off. Cross this one off, cross this one off. Exactly, it's written there. All I need to care for you to know is that lymphoid stem cells give rise to lymphocytes. Red blood cells, we're going to get a little more detail into that process, probably when we get back. But the white blood cells, I'm just going to narrow it down. Now, myeloid stem cells. Be very careful. It's easy to want to say lymphoid stem cells create all of them, but keep in mind they only care about lymphocytes. Myeloid stem cells will turn to the last remaining ones. Your monocyte, neutrophil, eosinophil, basophil, platelets, and red blood cells all come from myeloid stem cells. So what do we need? All you need to do, and I'm going to go ahead and make this easy to scale here, draw with this, make this larger. You can, for everything here, you don't have to know this. You do not have to know this. And try your best to mark that on your papers. This purple box, up to here. What you just need to know is that myeloid stem cells give rise to monocytes, neutrophils, eosinophils, and basophils. And don't forget about our platelets. This one we must know. This is that huge cell, megakaryocyte. These are where the fragments originate, they break off, and then they flow through your blood looking for any nicks on your blood vessels. And this is a process called erythropoiesis, which we're going to talk about a little more in detail next week when we get back. But today, what you need to log away is the myeloid stem cells give rise to red blood cells. You're going to see that we're also going to discuss next week something called the lymphatic system. And these are going to come up then, thymus, spleen, lymph, and we'll tie it with how blood is in plasma is directed or directly works together with your lymphatic system. So we're going to keep that note there. It'll be more important next week. And next, red blood cells. When we look at their overall profile under a microscope, this is probably what you saw. You can see all these little platelets. We're not seeing any white blood cells in this one. And you may notice that there's this little indentation or this little gap here in the center. And if you zoom in on them, what you see is that they have this concave disc shape. This shape provides a few things. It increases the surface area to volume ratio, or even to understand it more simply for us, is that it enables optimal, let me write this down, optimal hemoglobin carrying capacity. This word next is pronounced rolu, even though it's spelled like that. Rolu. And these are stacks. In some places, your red blood cells travel in a linear order. And so that's what you're seeing here is that some capillaries that they actually travel. They're traveling through very tiny little capillaries, and these little capillaries are so small that red blood cells go in one at a time. But for the most part, in blood vessels, it just looks like a massive amounts, millions of cells just sort of flowing through your blood vessels. But capillaries are so tiny, those exchange sites, that blood cells travel one at a time. And this shape also contributes to flexibility. Flexibility. And so that's what the functional aspects of red blood cells, which is attributed to that shape, gives those three properties there. It allows them to be easily stacked, very flexible, and it provides a great oxygen carrying capacity. Red blood cells, they lose a lot of their organelles because, again, they don't really have any other role than to carry around oxygen. There's no reason for them to create mucus. There's no reason for them to create proteins. Their job is just to carry oxygen and CO2 in reverse. And when they mature, they completely become anucleated. When you add an A in Latin to everything, it means the absence of something, right? A moral person is someone who has values and maybe some vices, depending on what culture you come from. But moral and then amoral means to not have any morals. So the word a, adding it in front of anything in Latin as far as the class is concerned, typically means it's absent, it does not have it. It lasts 3 months, 120 days. And this, you do not need to worry about this concentration here, but hemoglobin is the main component of red blood cells. In other words, there's a little tiny protein here, a little carrier protein that carries around oxygen. And that's called hemoglobin. And our next slide. We are almost done. I'm trying to go slow because I felt like the packet wasn't that long today, and I feel like we're still going to end early. And also, these are new notes, so I'm still a little clumsy with them. All right, so with that, oxyhemoglobin, right, has oxygen, right? We don't have to read all this to understand this. Oxy and deoxy means to not have any oxygen bound to it. This can contribute to some skin or color pigmentation that can be physically observed. And so when it's very oxygenated, when blood is very oxygenated, it means that it's usually going to be bright red. If it's deoxygenated, and I'm emphasizing those words because it's going to matter when we talk about blood flow next week. Deoxygenated means to not have oxygen. Oxygenated means to have it. If we examine hemoglobin itself, remember that when we learned about protein structure, we had alpha chains, we had beta chains, and then you have two alpha chains. You can see chain 1, chain 2, and then we have a beta chain and a beta chain 2. These are amino acid sequences. They're all together, linked together by peptide bonds, making a protein, a carrier protein. And within that protein, there's this little active site, this magic site, right? This is called the heme. Each one of these subunits contains one heme. One hemoglobin molecule has 4 heme groups. We're just going to write 4 hemes. And a heme group, if you examine it, what it's based on is iron. That's all that hemoglobin is that in this very center, there's a little bit of iron because iron and oxygen have a really good relationship together. What happens to an iron bar that you leave out by the beach, for example, and it's just there for a few years? What happens to the bar? It rusts. That's because iron and the oxygen around it interacts with it. They interact really well together. And so iron loves oxygen. It can bind oxygen and also CO2. And so iron becomes the main player of heme for the ability to hold onto oxygen. That's why some of you maybe have to take iron pills because you're anemic, and that's because you have a deficiency in iron. And it could be genetic, it could be a liver problem, it could be just not eating very well, it can be stress, a few things can cause anemia or the inability to really transport oxygen very well. So when some of you may have seen someone who says they're anemic and they got to take a pill, it's typically because they're replacing some iron in their body. And lysis, we see this word again. Hemolysis means when a red blood cell ruptures and is destroyed. And how long do red blood cells live for? 120 days or 3 months. 120 days or 3 months. And this is something we're going to learn about next week when we're going to talk about the formation of red blood cells and the death of red blood cells. And so we're going to put a pin on this. And erythropoiesis, again, becomes that same topic as to how do red blood cells form. I really wanted you to know the details of it. It's very useful information. And this, again, it comes from myeloid tissue. All red blood cells, you don't have to scroll back, but all myeloid stem cells, right, give rise to red blood cells. That's all that fact is saying. We're going to learn what this is next week with a picture, a really nice infographic I found. And so to be continued for those 2 points. And then to finish up the class, we need to talk about our white blood cells. When we look at our red blood cells, we have our white blood cells here, these large purple ones. This is the color that they stain, and that's how they appear in a microscope. And so when you, and then when you see here, you'll have 2 families. You have granular and agranular. And granular, okay, is going to imply granules. That's simple, granules. If you look at your illustration, or at least my screen here, you can see that they have these tiny little granules, hence the name granular leukocytes. And I always remember I like Neb, it always just stuck with me, but a lot of students like Ben, backwards. And this order that you're seeing is telling you the abundance as well, with neutrophils being the most abundant and basophils being the least abundant. Yes. And neutrophils, eosinophils, this is pronounced oo. Let's see, how can I, no, with double O's there. Eosinophil. This sounds more like an A to think. Eosinophil. And here we go. Eosinophil. I think that's what I had. Eosinophil. And then basophil. Basophil. Agranulocytes, what happens when we have an A in front of something? Absent. So it's absent of granules, and monocytes and lymphocytes don't have many granules. I'm going to be honest with you, I'm pretty sure they do have granules, like 100% sure, but they're not visible off of a microscope, so they're categorized slightly differently. But they do contain some granules, I'm pretty sure. And so this acronym will be a great way to remember the abundance or the order. Never let monkeys eat bananas. Okay, this is the order of abundance, with N being the most abundant: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. But be careful because I'm mixing them, okay? This is the granulocytes and the agranulocytes together. But you're seeing that now we didn't separate Ben. See, Ben is not together anymore. They're telling you by abundance. That's what this is supposed to help you with. And so, our granular leukocytes, 50 to 70% of our leukocytes are neutrophils. We have to know this value. Put a star next to it. Eosinophils are going to be in second place at 2 to 4%. And basophils are going to be less than 1%. Now their function. You don't need to know much about this. This is just a nice little reading to like sort of, this gives you a little description how it appears when it stains, because some of you might go into hematology, so I thought these notes would just be nice to have handy when you get there. And then these are just a little fun read, but what you just need to know is the neutrophil, the name, and the function. That's all you're going to need. So maybe write one more asterisk here, and don't let this table intimidate you, or don't waste too much time on it. You just got to worry about the functions. And these percentages are also written right here for you, so you can make a little flashcard out of this table, or you can practice by covering it. And so neutrophils being the most abundant, best way to remember what they do is that if neutrophils are the most abundant, you have to think about bacteria. Some of us have a general idea that, if you put a bacterial animal into a species into a something with agar in it, right? Like it's food. If it's placed in there, you can expect that after an hour, that one bacteria can become 1 million. And so what do we need more of then? What more of these little soldiers protecting us? Which one needs to be the most abundant? It's the one that needs to kill or attack bacteria. So they neutralize and engulf pathogens, specifically bacteria. They can do other things like viruses, but what you just need to know is that they engulf pathogens. And typically we're talking about bacterial pathogens. Eosinophils. Eosinophils attack parasitic worms. And here it is, parasitic worms. Parasitic worms. I say worms, but because most of them are worms, but otherwise that's not necessarily true. Parasitic infections, pardon. Parasitic infection. Basophils, these are going to be responsible for the release of histamine, and they promote inflation. They promote inflation. You want to learn this today. Histamine causes inflammation. So does anybody have an idea or seen it on the Claritin bottles? What are they called? Antihistamines. It's to reduce inflammation. So you can see that they're sort of to combat these overly sensitive ones. This is the reason that pollen may give you an allergic reaction. It's because of them. They recognize things, sometimes foreign things, and they say, this is dangerous. And then they elicit their attack against it. And I'll talk a little about that because we're almost done. So when you think about, let's say you have an allergic reaction, right? And you have all this, what happens to when you allergies? What's going to be happening to your nose, right? All those mucus, you can't control it. And that's because mucus in and of itself can neutralize pathogens. It's sort of drenching, clenching, or removing, sort of cleaning out your airway, wherever it detected that pathogen. It just wants to clear all that space out because it detected something foreign. And that's what these cells are responsible for, is detecting foreign things and getting rid of it. And so we come back to our eosinophils. They do the opposite of our basophils. So they reduce inflammation. Sorry, I've been reading this thing and I keep saying inflation every time I say the word inflammation. And then this is enough for us to even understand now. Engulf antibody-labeled materials. Remember that we said earlier that when something dangerous is in your body, antibodies will mark it, be bound to it, and then our eosinophils are the ones that come in and do the cleanup work. They'll get rid of things that are marked by globulins or marked by antibodies. And the last two that we need. Our monocytes make up 2 to 8% of our leukocytes. Monocytes. And then you want to write macrophages. And these I'll show you what they do with a better illustration for the second part of these notes when we get back. And then we have our monocytes, lymphocytes. Monocytes are going to be tissue specific, meaning that when you look at monocytes, they're made in one place, which we're going to learn about also next week on Thursday. But they're made in one place, and then they travel to the liver. They may travel to the kidneys. They may travel to the lungs. Some of them may travel I'm out of organs to really give you examples. I think you get the gist, is that every organ will have monocytes that travel to it and monitor it. And that's what this word means. That's why this word we should add here, they sort of turn into, monocytes, they turn into tissue-specific macrophages. And then they stay there, and then they become kidney defenders, they become lung defenders, they become a defender of whatever organ they're going to inhabit. But they start off as monocytes. And then when they arrive to their tissue, they just sort of transform into that tissue-specific macrophage. And lymphocytes, they produce globulins. They produce your antibodies. And that's how they provide defense. And now you need to add this note because I'm not seeing it here. Produce antibodies. All right. We'll end there, and then we're going to start off on erythropoiesis. So if you log a lot of this away, you've done a lot. And then because the heart is fun, I think it's easy to learn, not the physiology of the heart, but the heart itself, learning it is pretty fun and easy. Get this out of the way because this exam sneaks up on us. I'll give you the date and an announcement as soon as I look at the schedule. Okay. All right. With that, I'll let you all a little bit earlier today. And